Rosalind Franklin was a British chemist and X-ray crystallographer whose research contributed significantly to the understanding of the molecular structures of DNA, RNA, viruses, coal, and graphite. Although her work on DNA became widely recognized after her lifetime, her scientific career encompassed a broader range of research areas, each marked by careful experimentation and analytical precision. Over the course of her life, she established herself as a rigorous and innovative scientist, working in laboratories in the United Kingdom and France during a period of rapid development in physical chemistry and molecular biology. She was born on 25 July 1920 in London, England, into a prominent and socially engaged Jewish family. Her father, Ellis Franklin, was a banker and a teacher at the Working Men's College, and her mother, Muriel Whaley Franklin, came from a family with a tradition of public service. Education and civic responsibility were central values in the Franklin household. From an early age, she demonstrated strong academic ability and a clear interest in science. She attended St. Paul's Girls' School in London, one of the few schools at the time that offered girls instruction in physics and chemistry. By her mid-teens, she had decided to pursue a career in science. In 1938, she entered Newnham College, Cambridge, where she studied natural sciences. At Cambridge, she focused on physical chemistry and completed her studies in 1941. Although women at the university completed the same coursework as men, they were not formally awarded full degrees at that time. Franklin later received her degree retroactively when the university changed its policies. During the Second World War, she contributed to research that supported the British war effort. She joined the British Coal Utilization Association, where she investigated the physical structure and porosity of coal and carbon. Her work helped clarify how coal transformed into coke, an important material for fuel and industrial processes. Franklin's research on coal led to a doctoral degree from Cambridge in 1945. Her thesis examined the microstructures of various forms of carbon and coal, establishing relationships between their physical properties and molecular arrangement. This work provided practical insights for industry and laid a foundation for her later expertise in structural analysis. After completing her doctorate, she moved to Paris in 1947 to work at the Laboratoire Central des Services Chimiques de l'État. There, under the direction of Jacques Mering, she refined her skills in X-ray diffraction techniques. The time she spent in France proved formative. She became proficient in the use of X-ray crystallography to study the arrangement of atoms within solid materials, and she published several papers on the structure of carbon. In 1951, Franklin returned to London to take up a position at King's College London as a research associate in the biophysics unit, directed by John Randall. At King's College, she began working on the structure of DNA, a molecule already recognized as central to heredity, but not yet fully understood in structural terms. Using X-ray diffraction methods, Franklin and her graduate student, Raymond Gosling, produced highly detailed images of DNA fibers. Through careful experimental control, Franklin identified two forms of DNA, which she termed the A and B forms, depending on the level of hydration. One of the most significant outcomes of this work was an X-ray diffraction image later known as Photo 51. The image displayed a distinct pattern that indicated a helical structure. Franklin's analysis of the diffraction data allowed her to calculate key dimensions of the DNA molecule, including the spacing between structural units and the diameter of the helix. Her notebooks and reports show that she was systematically evaluating whether DNA was helical and how its components were arranged. At the same time, other researchers were attempting to construct molecular models of DNA, including James Watson and Francis Crick at the University of Cambridge. In early 1953, Watson was shown Photo 51 by Maurice Wilkins, a colleague at King's College who had been working on related DNA research. The image provided Watson and Crick with information that supported their model-building efforts. In addition, a report summarizing Franklin's measurements was shared with them without her direct knowledge. Using this data, Watson and Crick refined their model of DNA, proposing the now well-known double helix structure. In April 1953, 3 papers on the structure of DNA appeared in the journal Nature. One paper, authored by Watson and Crick, described the double helix model. The other 2, authored separately by Wilkins and by Franklin with Gosling, presented experimental evidence, including X-ray diffraction data, that supported structural interpretations. Shortly before these publications, Franklin had decided to leave King's College. In March 1953, she accepted a position at Birkbeck College, University of London, where she shifted her research focus to the study of viruses. At Birkbeck, she joined a research group led by J.D. Bernal, a pioneer in X-ray crystallography. There, Franklin applied her expertise to the structure of tobacco mosaic virus and other plant viruses. Her work demonstrated that the genetic material of certain viruses was arranged in a helical configuration within a protein coat. This research expanded understanding of viral architecture and contributed to the emerging field of molecular virology. During her time at Birkbeck, Franklin built a productive research team and collaborated with scientists in the United Kingdom and abroad. She traveled to the United States to work with researchers studying viruses, including at the University of California, Berkeley. Her investigations into the structure of the tobacco mosaic virus led to detailed models of its organization, including the arrangement of RNA within the viral particle. She also studied the polio virus, applying similar structural methods. Her publications from this period reflected a continued emphasis on experimental precision and cautious interpretation of data. In 1956, Franklin was diagnosed with ovarian cancer. Despite undergoing treatment, she continued her research and maintained an active publication record. Over the course of her career, she authored and co-authored numerous scientific papers spanning studies of carbon materials, DNA, and viruses. Her work was characterized by methodical experimentation, technical skill in X-ray diffraction, and careful analysis of results. Colleagues recognized her expertise in interpreting diffraction patterns and in preparing high-quality crystalline or fibrous specimens for study. Rosalind Franklin died on April 16, 1958, at the age of 37. 4 years later, in 1962, the Nobel Prize in Physiology or Medicine was awarded to James Watson, Francis Crick, and Maurice Wilkins for their discoveries concerning the molecular structure of nucleic acids. The Nobel Prize is not awarded posthumously, and Franklin's contributions were not formally recognized by the prize. In subsequent decades, historians and scientists have examined archival materials, laboratory notes, and correspondence to better understand the collaborative and competitive environment in which the structure of DNA was determined. These studies have highlighted the importance of Franklin's experimental data in the development of the double helix model. Beyond the question of recognition, Franklin's scientific achievements stand independently across multiple disciplines. Her early investigations into the structure of coal improved understanding of carbon materials and had practical industrial applications. Her DNA research provided essential empirical evidence during a pivotal moment in molecular biology. Her later studies on viruses advanced structural virology and influenced subsequent research into viral assembly and genetic organization. Franklin's career unfolded during a period when opportunities for women in science were expanding, but still limited. At Cambridge, she studied at a time when women were not granted full membership in the university. In professional settings, she worked in predominantly male laboratories. Nevertheless, she secured research positions, obtained funding, and published in leading scientific journals. Her trajectory reflects both the challenges and the gradual institutional changes of mid-20th century science. Her legacy has been recognized in various ways. Buildings, laboratories, and awards have been named in her honor. Biographies and historical studies have examined her life and contributions, situating her work within the